since Athens. Like Eduardo was telling something, I could not tell the same thing about Francesco there. And I used to work with TLDs. I still work with them. OSLDs, gels, um, films. But something was curious me, how the radiation really interacts with the body. So then I discovered the biodosimetry. So biodosimetry is crucial when these physical dosimeters are not available or insufficient, or you need a new approach. So today, my talk will present something, a model, a new model that we want to propose to you that is ethical and we can play. So it's in vivo, but we can play with this. We don't have ethical concerns. So what is biodosimetry? Biodosimetry is the estimation of radiation dose via biological effects. So we measure directly from the cells what the radiation does. But the traditional methods, the cytogenetic assays in human lymphocytes, are challenged because they are time-consuming, they have ethical concerns. I don't want to irradiate you to test anything. So we just use things that just have happened. So we are not testing things all the time. And has complex logistics. So we decided to do biodosimetry because it is important, even it's complex. So when physical dosimeters are used, they measure the physical dose, but they say nothing about how the body actually responds to that dose. What happens after? So individuals that are irradiated with the same dose can have different response. It's the same dose, but you develop cancer, and I don't develop because I'm more resistant. So these factors can be investigated with biodosimetry. We can use this to evaluate this process, repopulation, repair, reoxygenation. So things that we can do to complement the physical dosimeters. Both are very necessary. So let's take a look at the cell and remember a little bit what is this with the DNA. So the cell has a cycle. For example, most of cells in our body have a 24-hour cycle, subdividing in 4 phases. And the most sensitive phase to radiation is the mitosis, near the mitosis and during the mitosis. The mitosis is when the DNA double helix, it's concentrated to form another cell. And so the mitosis is this division. We have one cell that is split into equal one to the other. So you can evaluate the mitosis index, seeing the cells, which phase they are, and you can see in microscope they are in mitosis. We can recognize the mitotic cycle, and then we can measure the mitotic index, that are cells in mitosis, to the total number of cells that we see in our microscope. And we can see also if they are regular, so they are in normal phase, or if they have some defects. The most sensitive part of the cell to the radiation is the DNA. It's well known this. So what is the DNA? The DNA has this base. If you remember, cytosine, thymine, all these bases, all these bases that are really arranged in a sequence that gives you all the characteristics that you have. If you have long hair like me without curl, or if you have curl, it will be written there. If you will have some sickness, it is written there. So the cell has this perfect sequence. But when it replicates, it separates the two parts, the two helix. And how the two cells know how to form again these two helix? It's because one side has, for example, cytosine, the other side should have guanine. So when they are split, you have part here and part there. So to form the second part, the body knows it needs the complementary base. So when you have the other adenine, you should have thymine. So when you split this in the mitosis, you have two cells exactly the same as the previous one, unless this was damaged. And this can be damaged, for example, with oxygen radicals, the free radicals that we are use every day to take some oranges to avoid this. So this comes from the damage that oxygen radicals can cause. Ionized radiation, chemotherapeutics, this can cause damage, especially in one side, so single strand break. When we have high LET radiation or chemotherapeutics, you can have both double strand break. That's even worse. Now we have double problem. So we don't know what's the other side. When you damage here, you have the other side, and you know what is in the other side. But when you have a double, what is in one and the other? We don't know anymore. Our body doesn't understand and maybe can make something bad to us. So to kill the cell can be killed. If the cell is killed, it's okay. If it's not too much, not much cells are killed. But if it survives, it keeps the damage there. That is not so good. And when you consider the different radiation, for example, high LET and low LET, you know that it's worse to be exposed to high LET. But why? Because the radiation can cause the same the same type of chromosomal aberrations when it's exposed, but the frequency of these induced aberrations depends on the type and the dose of this radiation. So when you have high LET, you have more densely ionized. So it's more probable that this dense ionized radiation causes the double broken in your DNA. So you have a damage that is more difficult to be repaired. And so if the cell uh, is damaged, it can be repaired. Probably you already cut your finger and had damage, but your finger is still there because our body is prepared to repair many damages. So the mammalian cells, we are part of mammalians. So when it's suffering the environment, so more than 100,000 DNA lesions per day result in replication errors. But most of these are repaired. So our body knows how to repair this. So the radiation also induces many lesions, and most of these can be successfully repaired. So let's resume what we had until here. So we have the action of radiation that can be indirect or direct. Indirect is in the water. So you have mainly from low LET ionization of water around the DNA that form free radicals that damage the cell. So this, when you are exposed to X-rays, most of time, this indirect action happens. But can be direct action, can be direct over the DNA. So you have most probable when you have high LET damage in your DNA. And then the cell can repair this. So you have, again, a normal cell that can suffer mitosis, and it is completely normal. But when it's not correctly repaired, we can have mutation. And when the cell is mutated, it can die. As I told you, if it's not repaired and died, if it's not much, we can keep this cell safely. The cells that remain keep us alive. It's too much, we die, of course. But when we don't have repair, so this mutation continues, and the cell continues to mitosis. Now, instead to have one cell with damage, we have double, and continue, and continue. And this can be the cancer. So we can have this somatic infection in your body that must be prevented. And how can you know what is in the cell? So we can visualize these effects, this damage in microscope. So this DNA damage is visualized in chromosome as some pieces or something strange, like micronuclei. They arise when the whole chromosomes it's not complete inside where it should be. So it appears a piece of chromosome outside at the end of the mitosis. So the cytogenetic damage can be measured. So we can measure in the cells. For example, here's micronuclei. You see pieces that are not inside the cell. Here you see things that are not completed. So we can measure this and see the frequency. So we have this frequency of biological markers. And doing this in correlation with the total cells that are there, we measure the normal cells, we measure the damage, each kind of damage, so we can do biodosimetry. And then what we do? We put the frequency of these damages to the absorbed dose. And we can fit this frequency and see how it is the behavior. For example, the most used model is this linear quadratic model. The linear quadratic model describes how are these biological effects as function as the absorbed dose. So we have part that is linear and part that goes to the dose to the square. So we can see in our experiments that the frequency obeys this, so we can study this. You'll see more about this in Francesca's presentation after. So the interaction of radiation with a matter depends on the type of radiation, the density of the matter, the Z of this matter, but also depends on many factors after. We usually consider only this, so we have many things happen. So what happens after? So this is why we decide to study biodosimetry. But as I told you, we cannot do this in vivo easily. I cannot take you to do experiments. We have a really bad history about that. These children were fed with cereals that was radioactive, just to see if the nutrients were going in the body where they want. Of course, we cannot repeat this. And even animals. Can you imagine to irradiate millions of animals to know details how the radiation is behaving in your body, how it proceeds after the dose is absorbed? Of course, we have these concerns. And then we thought in something more ethical that we could use and we could play. I like to play that there is no save the onions yet. So we decide to use plants, especially the onions, to play with this, to irradiate and see what happens after the dose is absorbed. So we took allium cepa. Allium cepa is onion. So it is well known that allium cepa test works, for example, for chemicals in the environment, and they correlate the effects of this allium cepa to the effects of mammalian cells. And this is really well known that works. We can have a lot of information from allium cepa. So this is an efficient model to determine chemical pollutants genotoxic. So radiation would be one more. It's a special species that has a low number of chromosomes, a lot of good characteristics that we can observe. So the tip is transparent and the cycle is quick. So have a lot of excellent options to make the biodosimetry. And more than to study the damage, we can also study the protection. So we can study radioprotectors, so agents that reduce the harmful of the radiation effects. So we can put also this in the onions to see what happens. For example, when you irradiate X-rays, you have the formation of free radicals. If you remove the free radicals with something like C vitamin, you diminish the damage. So we can study new radioprotectors with our samples. We also can play with a photobiomodulation. So photobiomodulation is the effect of a red light or some kind of lights that reduce the damage. So how it works? We can play with this using these onions. And how we do? So we experiment with several kinds of radiation. we stop this cycle, and we look at the microscope to see what is there. We count this and we model. So here is our samples being prepared. They should be inside something that's like a refrigerator, but it's not. It's a BOD that it keeps in determined temperature, and then the seeds grow, and you have this meristem. So you used to irradiate this part. We can irradiate. Here is a beta source, is a linac. So we can use different kind of dose, different kind of equipments. We can play with many things, and then we can stop the cycle. We stop the cycle with chemicals. We put this in something that gives color, so it becomes red. And then we put this sample in the microscope and look what is there and count the damage. So we compare with unirradiated, and we can estimate what is the dose effect. And we repeat this a lot. So 1,000 cells per slide. This is called the slide, so we don't know what's happening there to not be induced by this knowledge. And we repeat at least 3 replicates per each dose. And this is a kind of a result we have. You see, this is 2 micronuclei, 1 micronuclei. We have counted this. Here you see another kind of defect. So we were counting these aberrant cells with damage, and we were putting this in the graph. So, for example, this is a result. We irradiate with beta, and we have the response with 1 micronuclei, 2 or 3, or the combination, and we can modulate this, have a model to explain what happens with radiation, with the cells irradiated. Here, for example, is the response to the UV radiation. And we have linear behavior, so we can estimate the dose that somebody received if we have this onion exposed in the environment, for example. Here is the dose response of micronuclei induced by alpha and 6 MeV linac. What do you expect from different labs? You expect that high LET alpha produce 20 times more defects, more damage than X-ray. And it's exactly what we got here with our onions. So it's a good model to study this. And more details you can find with Francesca after in this section. We also are studying the radioprotectors. As I told you, we can use a different substance to scavenge the free radicals. And we are doing these experiments in the lab, put some different kind of like India ficus is in the water and growing the sample and then radiate to see if there is damage. And you can find more details about this in the poster on Thursday when Tommaso and Francesco will present this. And also the photobiomodulation. We are doing the experiments in the lab. Even the onion doesn't have the same kind of absorption as our body has, but we can try to make some experiments to see how photobiomodulation repair the damage. And we have been doing this, and we saw that really the light can diminish the damage, and we are evaluating this. For example, here, higher the dose, higher was the effect of this light diminishing the damage. And this, you can find more details with poster with Vinicius on Thursday. So, in summary, the potential of a micronuclei analysis with allium cepa was verified as a sensitive support system for detecting dosimetry as screening genotoxic effects induced by low and intermediate doses of different types of radiation. The investigation with a great variety of radiation type, radiation protectors, photobiomodulation, everything is in progress. We invite you to collaborate with us to determine this in meristem allium cepa. And we have published several papers that are available. So I invite you to look more details in these papers. And I would like to thank especially my students. Without them, I will not do anything, not mine only, but in Italy also, the collaboration and the financial support from our government agents, CNPq, my local agents, FAPTEC, and CAPS, that is national in Brazil. Thank you very much.
Very good, perfect timing. We do have time for questions for Susanna.
It's very different.
Oh, I have
one. Thank you, Susanna. I learned a lot from your presentation. As I understand it, allium cepa has been used for a long time as a surrogate system for looking at radiation effects.
But high dose, they were using for high dose. Low dose was never.
For low dose, okay. My question was, do we know how well what we learn in allium cepa can transfer to a human?
Yeah. It's not only studied to radiation effects, but also to pollutants. It's very well understood how to transfer these phenomena, what happens in onion, to the human cells because of this knowledge in pollutants. And it is similar, the results up to 95%. So 95% you and me are onion. It's almost unbelievable, but the results seems to be really useful.
Thank you very much, Susanna, also for that last statement, which I very much enjoyed. Using artificial intelligence to look at the micronuclei dicentrics and then all the different potential results, could that give us another dimension and more layered information to look at damage?
For sure. I have one student that knows how to use artificial intelligence, and he's trying to help to get information. But please, I want more people. This is difficult to find people that knows how to work this and extract information. But yes, and not only to get different kind of information, just to count. It's difficult to count. Imagine you stay there 2 hours, 3 hours. I was never doing this. They are Francesca is doing this. So they are there after 2 hours counting. It's very tough task. So they are tired. So if you can count with some program, this would be much more easy. But also to have information how to conduct. For example, we are just measuring most of the samples only 24 hours after the irradiation. We irradiate, we wait 1 cycle to have mitosis, and then we measure. But we can wait 2 cycles, 3 cycles. We can play a lot. We just need more people to do.
Otherwise, we'll be running behind schedule.
Thank you. Thanks for the nice presentation of the correlation of the physics and biology. Did you also go into studies? Do you have data on those rate dependence?
Yes. Yesterday I had one paper accepted on those rates, just yesterday. Yeah, we are studying, and we want to study in flash also. We want to do that in Italy.
Thanks again.
Thank you.